Hi, my name is Raimah Hossain, and today I'll be sharing my research regarding use of the novel method of the Feammox pathway to treat domestic wastewater. Treated wastewater is typically delivered to our local rivers, so it can have implications on both the environment and human health, which is why this process is so important. So, for example, if there's an abundance of nutrients left over in wastewater, it can lead to large algal blooms that deprive aquatic ecosystems of the resources that they need to survive. My research specifically focuses on anaerobic biological wastewater treatment as it is more energy efficient and costs less than aerobic wastewater treatment. Specifically, iron or ferric dosed anaerobic technologies are really useful because they can utilize waste such as acid mine drainage sludge that is high in iron to fuel the process. As seen in this figure, ferric reduction can be coupled to two different pathways in ferric dosed anaerobic technologies. One is the organic carbon oxidation pathway, and that bottom pathway is the ammonium oxidation pathway, which ferric reduction coupled to ammonium oxidation is what we refer to as the Feammox pathway. Now, since domestic wastewater contains both organic carbon and ammonium nitrogen, both of these pathways will likely coexist in any wastewater treatment system. But the organic carbon oxidation coupled to the ferric reduction is generally more thermodynamically favorable than the Feammox pathway. And there's a lot of conflicting research findings on whether the presence of organic carbon will inhibit or promote the Feammox pathway. And having all this conflicting and unclear information, the purpose of our project is to really explore the effect of organic carbon on the efficiency of the Feammox pathway, specifically in removing nitrogen from wastewater, so we can assess its potential for real world application. Now, the way that we really assess the effect of organic carbon on the Feammox pathway is through our experimental design. So as you can see, figure 2 and figure 3 employ pretty similar mechanisms. There's an influent synthetic wastewater tank and an influent ferric chloride reservoir, which flows through the bioreactor into the effluent water. The influent synthetic wastewater is the only thing that's different between the two figures, as figure three's wastewater contains both ammonium and carbon as opposed to just ammonium like figure two's. Now, how do we assess the effect of this presence or absence of organic carbon on the Feammox pathway? We take samples of the effluent water and we analyze it using UV vis spectrophotometry to analyze that ammonium or nitrogen removal percentage and the organic carbon removal percentage. We also use qPCR or quantitative polymerase chain reaction to see how much Feammox bacteria is present in the sludge of each bioreactor. Now, as we mentioned, the ammonium removal percentage can be seen here over about a 180-day period. The reactor without organic carbon shows an increasing trend in removal percentage, and at the end, it's about 20% higher than the reactor with organic carbon. The organic carbon removal in the reactor with organic carbon reaches almost about 100% over that same time period, showing that that organic carbon pathway is likely occurring in that reactor. And then finally, we'll see the quantification of Feammox bacteria measured through number of gene copies per milliliter in both reactors at 3 different phases. It's important to note that I was only involved in the last phase, but that all 3 phases were shown. So you could see the increasing trend in the reactor without organic carbon and the decreasing trend in the reactor with organic carbon when it comes to the quantification of the Feammox bacteria present. Now, all this data really just helps us form the conclusion that helps us shape future biological wastewater treatment systems. The qPCR results suggest that the absence of organic carbon is what actually promotes the growth of Feammox bacteria over time. And our ammonium removal percentage data just further corroborates this by showing that the reactor without organic carbon has the higher ammonium removal percentage and the increasing trend over time. In terms of continued research, it's crucial that further study of the microbial community is conducted so that we can understand all the concurrent pathways occurring within the bioreactor. It's also important that a two-stage process be discussed since our results indicated that high organic carbon presence in domestic wastewater may inhibit Feammox activity. In this first stage, organic carbon would be removed, and then in the second stage, the Feammox pathway would be applied. I'd like to thank you all for listening to my presentation, and I'd like to thank the SURE program for its research funding.